Professor Ode, we have just heard that biofilms are responsible for health, but also for diseases in humans. You research these comrades in arms of our health. The focus is also on pathogens that frequently occur in hospitals, so-called multi-resistant germs, which are also increasingly limiting the effectiveness of antibiotics. What do we know about them today, and how can we counter them wisely? Because if we get them more and more antibiotics, I think they find more ways to get resistant against them.
Yeah, so thank you very much for this important question. I think so it is very easy to discuss the importance of infectious diseases by focusing on antibiotic multi-resistance. I think that multi-resistance and the emergence of multi-resistance is one facet of infectious diseases, and the fascinating aspect of biofilm-associated infections is that these infections occur and persist, even very aggressive antibiotic treatments, despite their ability to express specific resistance mechanisms. So it's the ability to form a biofilm per se that induces this, what we refer to as phenotypic resistance. So if we talk about preventing infections related to multi-resistant bacteria, so there are some very evident ideas like interrupting transmission routes, reducing the exposure against antibiotics. So these are very valid strategies. But I think, and this is very important to note, that the infections that we talk about, these biofilm-associated infections, they occur irrespective of our antibiotic consumption, because this is an inherent ability of the bacteria that will cause biofilm-associated diseases. So the staphylococci from the skin, they have been living on the skin for hundreds and millions of years, so they have been adopted very strict to the host. And probably biofilm formation is also important to colonize the host. But so as soon as we open up the window by introducing artificial surfaces, implants, etc., this inherent ability of these bacteria that are very important for human health turn into something bad. So they, that's why some colleagues refer to like these biofilm infections as infections caused by accidental pathogens. So these pathogens not necessarily are aggressive, and that is why it is so difficult to employ like the standard principles of infection prevention to biofilm infections. So here these the idea to reduce antibiotics consumption and antibiotic exposure doesn't work. So we need more sophisticated ways to prevent biofilm formation, to prevent colonization of indwelling medical devices. And Meta just referred to this tremendous number of infections, and that is my last point that I would like to make. So if you look at the number of patients suffering from infections caused by multi-resistant bacteria, these are clearly outnumbered by those patients suffering from biofilm-associated infections, which don't necessarily have anything to do with antibiotic resistance. So antibiotic resistance is an important aspect of acute care medicine, but these biofilm infections, they are more like an overarching principle of many chronic infections.
Does that mean that if I wash my hands too often, it can bring bacteria out of the balance, and then I get an infection?
Of course. So I think it's very important to know that hand washing doesn't necessarily puts you at risk for infection. That's important to note. But it is also important to acknowledge the fact that everything that we do, every exposure against chemicals, antibiotics, whatever we do, this will have an impact on the colonizing flora of the human skin. And of course, this does not necessarily lead to disease. But if we have like additional predisposing factors, then the sum of all these disturbances can gradually increase the chance to develop disease. Exactly.
Oh, thanks a lot till now. I think we come back to that. Professor Yildiz, you are an expert, as said, on the cholera pathogens and their effects. As I said in the beginning, up to 4 million people worldwide contact cholera every year. If it left untreated, they die very quickly, and it's about 140,000 people who died on them. What role do biofilms play in this illness?
So, Vibrio cholera, many pathogens, human pathogens, actually live outside the environment of human host, and Vibrio is one such pathogen. So it can grow in aquatic environments. It can form biofilms on many different surfaces, and when it grows as a biofilm, it can have access to nutrients that accumulate at interfaces. For example, it can form biofilms on chitin surfaces, then while growing on these surfaces, actually, it has ability to kill other bacteria and acquire their DNA, and they can evolve. So it helps to evolve, it helps to grow, and when grown as biofilm, it's also resistant to disinfectants. For example, chlorination is a first means to kill pathogenic bacteria, but when grown as a biofilm, Vibrio is resistant to chlorine-mediated killing. Humans acquire Vibrio by ingesting this pathogen through contaminated food and water, and biofilm bacteria are actually producing more of the cholera toxin, which is responsible for causing diarrhea. So they are primed to do infection. Furthermore, as organism goes through the gastrointestinal tract, it's more resistant to acid-mediated killing in the stomach, because that's the primary defense. And it can also survive better to all the human defense systems that would kill bacteria. And when it comes out as a biofilm, it's also more infectious. So it could facilitate human-to-human transfers during endemics, during outbreaks, but it can also facilitate further survival in the environment. So it's really very important for all aspects of its life cycle: growing in the environment, evolution, antimicrobial resistance, and increased ability to cause disease. So it's part of every aspect of its infection cycle.
Do they have a better chance to get into humans if they build biofilms? Is that better for them?
Actually, it's better for Vibrio, but
yes,
not for humans. So,
yeah, well, if I would be a bacteria, would it be nice if I build a biofilm?
Yeah. So actually, we know that when they grown as biofilm, chances of transmission to humans is greater. There are field studies where people use in cholera endemic areas, when they use sari, clothes, to filter the water, transmission rate is reduced by 50%. So you are dealing small, 20 micron size particles. If you can filter them, you can reduce infection rate quite a bit. So biofilms helps to be transmitted to humans, and it grows very rapidly within humans. Like a patient with a full-blown cholera can lose 20 liters of stool a day, and they can excrete a huge number of microorganisms to environment. If they are part of the biofilm, they're gonna survive better. Amplification rate is greater. Yeah, and I just want to say, actually, they grow on marine snow. For example, you indicated that you studied these organic particles. These organic particles are really buffet for them. You know, they can get quite a bit of nutrients.
Okay, so they are quite interesting for you because they are so ubiquitous.
Yep, they are ubiquitous, but also it's interesting from fundamental biology aspect of it. Like Mattia say, you want to know interesting biological question, and one can learn a lot from basic biology by just studying biofilms for sake of studying biofilm without considering any environmental, industrial, or clinical application. Because that's where you do the discovery. Like basic science aspects of it is very interesting to me.
Thanks. Professor Sondermann, till now we have heard a lot about biofilms and disease. But biofilms could also be our aliens. Professor Landau said that, eating plastic, for example. But there's another example which can be done already if we apply a special fluid with lactic acid bacteria and partners to the walls after cleaning and disinfection an animal stable, for example, chicken stable, then they spread so quick that the wet ones don't have any chance. So is there any other positive effects on biofilms?
Right, so that's a great question, and I think it takes back to what Metal said in the beginning. Biofilms are really everywhere, and this is something what my revelation when I grew up was that I was trained that bacteria are, compared to us humans, fairly dumb organisms, single cell, moving around, not doing something interesting. And I think now we appreciate that biofilms are actually the preferred lifestyle of bacteria. Most bacteria actually like to sit in biofilms and act as communities and not be alone. They can change the life cycle and they can switch, but really the preferred lifestyle is in biofilms. Having said that, biofilms are also everywhere. So if you look around, you actually see them, you can find them everywhere. A few good examples are, for example, bacteria colonizing in soil as biofilms or living in soil as biofilms, colonizing, sitting on roots and helping plants basically to metabolize nutrients more efficiently or protect the roots of plants from drought, for example. So this is a natural example where biofilms kind of are helpful, where we don't need to interfere actually. So this is just natural environment. There are other examples that Metal mentioned at the end of the introduction where we can actually use biofilms for good. And this is, for example, bioremediation, like cleaning of wastewater, cleaning of oil spills, applications that benefit from bacteria growing in biofilms where they actually applied as biofilm cultures in bioreactors. Another sector is natural products that are unique to bacteria growing in biofilms. Those are kind of very unique sources of new natural products that could include new antibiotics, for example, for the bacterial warfare that basically was also mentioned this evening already. So there are many examples where biofilms are actually good. When we talk about hosts, in this panel we have heard about hosts so far as the patient that is infected by a pathogen. But host can be also something good, like a symbiosis. In the ocean, there are many sea creatures that are actually living in symbiosis with bacteria, and this is a good thing. They need this for their life cycle. The bacteria need this host, but the host also depends on this on the microbe living in them in biofilms. So those are natural systems where we encounter biofilms as well.
And you said they are helpful. They could be helpful. Have you an idea how we care that they stay helpful? Because sophisticated ways, I already heard that word. So is there a sophisticated way that I say them, okay, please be nice to me and to the environment?
Yeah, this goes back, for example, to antibiotic treatment. When we take antibiotic, we often wipe out our microbiome. And seeding a new microbiome, I mean, you take probiotics, but as soon as you stop eating yogurts, those bacteria often kind of are shed as well, and you do not develop a healthy flora as quickly as you want. And there are approaches where people actually harness this power of engineering and designing biofilms to actually reconstitute your microbiome in your guts, for example.
Okay. So, Professor Landau, when you heard all this and you get the introduction, what do you think, what is the most interesting idea in the ground research, on the basic research right now? Would you take them, please, thanks.
For me, it's interesting how they build the structures that are so strong that they can leach on and they can even survive really high temperature. When you go to a dentist and they put this to sterilization, you don't get rid of the biofilm even if you get rid of the bacteria. And my personal interest is how the material that makes them so stable is similar to what we have in the brains of Alzheimer's patients or Parkinson's patients. So for me personally, this connection between biofilm and neurodegenerative diseases is a point of research from the evolutionary perspective, from the applicative perspective, and from the chemical perspective as a basic scientist, is how you can form such stable structures.
So you say the structure by Alzheimer is the same as biofilms? Looks like the same?
They have very similar chemistry from what we know so far.
Okay. So the idea is once I tell them, be nice to me in my head, and then I don't get Alzheimer?
Not necessarily, but my hopes, I don't know if it's far-fetched for the future, is that we can take some kind of probiotics during our life and then we will not get Alzheimer's.
Okay, that's a fine idea. What do you think, Professor Rode? Would that work?
Well, that's an excellent idea, of course. But I think that it's not an easy way, because what we understand is that from the perspective of a clinical microbiologist, we see that almost every bacterium can cause disease at one point. And so just by the idea just to add a good bacterium to remove all the bad, that not necessarily prevents infection, because this so-called good bacterium can cause disease by itself. So we really need more information, and we need to better understand how these bacterial communities, and probably they are also like forming biofilms on the skin, and they are for sure forming biofilms in the gut. So we need more information how these good biofilms are really stabilized. And I think so Meta's work is going into an important direction, because we need to understand how these bacteria really can form these tethered aggregates, and if we understand how this glue functions, then we probably can also use this information to establish new models and or good biofilm communities that would also colonize the skin or the gut. But I think it's a long way to go and a hard road to travel right now. But we are only at the beginning of understanding how these within host biofilms are being controlled, regulated, so the host will play an important role in the immune system, how the immune system really interacts with these biofilms. So the idea is straightforward, and it's a brilliant idea, but I think that there will be a lot of pitfalls that we will have to encounter.
Professor Yildiz, what do we know about how they communicate with another? How shall I imagine what they do? Say they, hey, would you have a cooperation with you? Or how do they cooperate? How do bacteria communicate? I'm sorry.
Yeah, I think it depends on how you say it. You know, so, chemical signaling, right? Chemical communication, and the bacteria release chemicals to communicate with one another. But we also know that they can sense the touch, right? Mechanosensing is important part of signaling that bacteria uses to perceive that they are on a surface, right? And when they touch a surface, they can actually, they have appendages, structures on the cell surface, and they sense they are on the surface, they can retract, shrink that appendages that would bring them to a surface, then they can start producing glue, right? That's one way of communicating. I don't know, was there any other communication?
Okay, that's the way they communicate.
But chemical and mechanical signaling, basically. Yeah.
Okay. So when I look at you, when you're talking to us, I think you are really fascinating by that, are you?
Yeah, I'm always, little things fascinate me.
Well, because when I first read bacteria can communicate, I know it has been about 20 years ago.
Yeah.
I was really fascinated because I'm a biologist myself, and so I was really fascinated. Said, okay, that's the way I can imagine that they come together, but that I didn't know that it's their lifestyle, Professor Zonderman. What means that? That normally they don't exist in a single version. They always look from partner and more partners.
Right, so what fascinates me is all the computation that a bacterium can do, and the things that Fitnat and myself are studying are basically the communication pathways, how basically cells sense their environment. Touch would be one, chemical signals the other one, nutrients, humidity, other animals, other microbes around them. So what really fascinates me is how do bacteria actually use that information and then basically come out with a physiological response to go into a biofilm, to become virulent or more virulent. And it's really interesting because the bacteria like Vibrio that can live in very different environments, like in mud puddles at 15 degrees, in us at 37 degrees, in our guts, they are very good in basically making those decisions, and they have a large network of molecules that talk to each other within the bacterium to make intelligent decisions. A bacterium that lives in a cell and its main purpose is to kind of invade a cell has usually much more simpler signaling pathways or pathways to basically make decisions because it's not dependent on sensing the environment. And this is really something that kind of fascinates me and us in our work, that understanding how decisions are made, what is the input and what is the output. I mean, what decisions are made, and the decision is not just binary yes and no biofilm, but it can actually be a very graded and very variable response.
So it may be that the biofilms in our gut control our brain? Okay.
Definitely.
Imagine that for a moment, please. You are not brain, you are gut. Okay, that's really fantastic because I think it opens totally new doors on seeing on things we always watched, not even infections, but also depression or something like that, which all to do with that. I'm sorry for that. So if you know so much about the way so much about the way they communicate, you have any ideas how we can influence this communication? Are there any strategies right now? Solomon?
Yeah, there are ways certainly how to cut off communication between bacteria. So there's one signal or multiple signals that tell bacteria how many cells are around me of my kind, how many cells are around me of a different kind. And if you could manipulate that, and there are kind of tools, I would say, right now developed to test this way of treating bacterial infections or controlling biofilm formations by cutting off communication. So that would be, for example, one way to do that. I think another way would be to look into materials that are more resistant to biofilm growth. So it's more an application on material properties than on communication cutoffs.
Professor Rode, you were thinking what you want to say. I see that you would like to say something.
Yeah, absolutely. And so I remember maybe 20 years ago, so when these communication strategies were unraveled in staphylococci, everybody was like thinking, okay, now we have this golden knife and we can really interfere with the language of this important bacterium. And intriguingly, so not only from animal work, but also later on in like some phase 1 clinical studies, all these applications turn out to be wrong because these signals, as soon as you interfered with these signaling systems in vivo, they turned out to have a completely different role compared to what we expected them to have from the in vitro lab systems. So I think that's why it's still, we are, although we are moving forward and we are understanding more and more about the language of bacteria, but still we have this gap between what we are doing in the lab and, at least in clinical medicine, what is happening in the patient. So that is really an aspect that needs to be addressed, and where we need more sophisticated models looking at these biofilm infections. And another thing is that like the language is so diverse. So it's not this one language that we need to understand, but probably all these different languages and how they interfere with each other. I think it's one of the most fascinating aspects in microbial or biofilm formation. And it opens doors, but more questions than answers. Fortunate enough, so because then we can.
Fortunately for you. Professor Landau, could you add something to that?
For me, the fascinating part is that they change from the lab to the patient because they communicate with our cells. And they communicate and they interact in a very surprising way. Sometimes you expect something, but the reality is completely different because we are designed to fight this bacteria, like immune reactions, but sometimes they kind of hijack the systems that we have. They imitate us, they take control of the immune system, but sometimes they're doing something that is surprisingly good. So for me, the bacteria, we cannot study them in the lab on their own. We have to study them in the context of interactions with us.
Professor Yildiz, you have animal models. I guess that you try to do just the bridge from lab to patient.
I don't know if I go that far, but we would like to simulate infection using different animal models. There is no perfect model, but presence of bacteria in the gut prevents Vibrio colonization. So we have to use animals that are really young, and we can give millions of Vibrio cholera and see how the infection develops. This organism causes diarrhea, so we see how much diarrhea is being produced, and we also look at how many numbers, how much bacteria is present in the intestine. So they've been very informative. You have to be able to model the infection, and again, sometimes we think that, oh, this protein could be important for infection process. Sometimes that doesn't turn out to be the case.
So bacteria is something like always surprising object of your research?
It is always fascinating. It depends on how ready you are to be fascinated, but there are, just to give you an example, the organism that we study has 4,000 genes. This doesn't seem like too much compared to humans. But we do not know function of those genes, like 75% of it, we have no idea. And this is true even for best studied organisms. So that means there is always a room to discover. And when you think about, like we are talking about Nobel Prize, CRISPR-Cas, or this type of studies actually came from studying bacteria and viruses in their natural concepts. So there is always something to be discovered. I mean, these are really recent discoveries when we think about it.
Professor Sondermann, you said that this can be very important for food design. So I know that the people who are dealing with yeast try to put them on the environmental or even in the genome of those. So do you think it's a good idea doing that at the time? When I listen to you right now, I think you were just looking and finding every day something new, that they do that already?
Probably, yeah, because the bacteria have figured out how to live in very diverse environments and actually are also evolving much quicker than we are, and bacteria are evolving quicker than fungi, than yeast, for example. So I think they are really good in colonizing different areas and actually exploring new niches where they like to thrive. And I think you find bacterium we find bacteria that degrade plastic, for example, which I find fascinating. Certainly not their natural environment, but they can do this. So I think, yes, there's new chemistries, but also new lifestyles.
Okay, and so you think we could put them out in environment right now without having any skepsis?
Many bacteria are already in our environment. Like if you talk about Pseudomonas aeruginosa, which is a pathogen in cystic fibrosis, very important, or burn wounds. This is a bacterium that lives in soil. This is not something that kind of humans have created or we need to put out there. I think engineering bacteria that do certain things, that is probably difficult to control, because if you go into the field, they basically can go into the environment, and that's the reason why we are still working in BSL2 labs, where we certify that these things do not leave into the environment. So I think one has to be careful.
Okay. Last question for you four. What do you want us to take home as a very, very good message? What shall we remember when we leave this room? You've got a minute to think about it and to find one sentence for that. Okay, the minute is over. Professor Sondermann, you can do that in a minute.
I think that bacteria are absolutely fascinating organisms that can do much more than we anticipated early on, and that they are really kind of very good in sensing the environment and reacting to it.
I hope your life may be full of good biofilms.
So I would say that although we are very much interested in the so-called bad biofilms, I think the most important thing that we really need to appreciate is that bacteria are more than one a single cell, that they form communities where you have the division of labor, etc. So they are far more complex, and probably they are more similar to eukaryotic cell systems as we have appreciated so far.
For me, I see microbes and in general as our best friends and our worst enemies, and I feel more humble against microbes when I get to know them. We think of them as dumb organisms. They're really not. And even altruism in bacterial community, they sacrifice some cells for the other ones, so there is a lot to learn. And I think a lot of the questions here on how we are going to treat biofilms, the answers are coming from learning how they do it, and then we are just imitating them.
Thanks a lot for the expert, and I please give him a big applause. Thank you to you for participation tonight. And one thing really got me thinking this evening, and you were the one who said it once more, perhaps bacteria, we could learn something about bacteria even for our social system. Do more communication, do more altruismic, and all of us will have a better life. I wish you a good way home and stay healthy, and as said in the beginning, care for your balance systems. And now have a nice time upstairs and good home. Bye.